Chapter 3. The Line Hypothesis. The most fundamental element of life is a molecule called the entanglement molecule, or EM. This molecule, composed of normal baryonic matter, manifests the unique property of prolifically establishing a natural teleportation channel, which is a shared quantum coherent state, a quantum entanglement connection, or QE, with a hypothesized form of matter called metamatter. Metamatter is composed of an undiscovered type of particle which necessarily resides entirely beyond this space-time, in Hilbert space, or the metaverse, if you will. Metamatter is as essential to life as dark matter is to galaxy formation. Entanglement molecules in this universe are at all times entangled to particles of metamatter in Hilbert space. It is their natural state to do so. Metamatter, as is possible with any natural entity having only subtle degrees of freedom within this space-time, is not subject to locality or relativistic constraints, and so, via this QE connection, is non-classically instantaneously accessible to entanglement molecules, or EM, everywhere in this universe. These entanglement molecules and metamatter are the Alice and Bob endpoints of each isolated, naturally occurring QE connection established within every living cell that has ever existed. An entanglement molecule, once arranged from its constituent atoms, not unlike the molecules in the ferrite magnet in a transistor radio, is instantly sensitive to available, uninstantiated QE degrees of freedom, or DOF, of the QE spectrum, or quantum entanglement frequencies, or QEF. It is the QEF that define the unique natural teleportation channel upon which to entangle available metamatter. Such isolated pairings existed on Earth for eons, and in this universe for even longer before the naturally occurring circumstances arose, on Earth and perhaps elsewhere, to provide a sphere of molecules that could be described as an early cell wall. Not all entanglement molecules were likely to encounter a cell wall, but those that did, enclosed by this barrier, obtained the benefit of an extra level of protection. This enclosure allowed them to develop beyond the typical. This basic entanglement relationship is the most fundamental manifestation of life. It establishes the position of view, or POV. Over time, other types of molecules joined with these protocells, sometimes to their mutual benefit, sometimes not. Those that added no benefit or diminished the protocell's survival prospects would not survive. The QE connection gave surviving protocells something very special. It gave the otherwise inanimate molecular components on the inside of this early cell a form of intracellular communication. That is, the ability to interact at a distance, but more critically at that point, the QE connection gave the protocell the capacity to share or imprint internal cellular state information upon its entangled metamatter. Metamatter, because of its extra-dimensional non-locality and relativistically unconstrained nature, essentially acts as a kind of cloud storage for information accessible instantaneously from any location in this universe, and in any other as well. This universal cloud storage repository of information is the critical factor required to get evolution started. This natural cosmic background, Bose condensate, or CBBC, is what makes being possible anywhere in this universe. At that point, evolution existed only via random environmental contact between protocells with other structures in the primordial environment of early Earth. Thus, the cell became nature's biological entanglement circuit. Each such entanglement pairing constitutes an instantiation of life, whether on Earth, elsewhere in this universe, or anywhere in existence. Consequently, life could now be hosted by any viable formation of cell or cells that may emerge anywhere in existence. Once instantiation is established at one specific QEF, a unique value of the degrees of freedom among the infinity of possible values on the quantum entanglement spectrum. A QEF that is unique in all existence to each individual and to no other, but only while that QE connection, one's natural teleportation or line channel, persists. These yet-to-be-determined DOFs perhaps frequency and others, on the Q-E spectrum is the singular property in nature that defines each living individual. All other components of the instantiation process may change or be exchanged, but it is your QEF that positions you as the central and only target of your instantiation, of your life, and not someone else's. Change or retune one's QEF enough, and you change the being, the individual. You are your QEF. You are not your cells or your metamatter. It is very likely that the Q-E spectrum predated even the Big Bang. Your QEF is the immutable, the classically indestructible you. When entanglement molecules, contained within viable hosts such as the cell, located on any viable planet, orbiting any viable star, anywhere in existence, entangles metamatter at your QEF, that is where you will instantiate. That is where you will be. A place such as that is where you are right now. A place such as that is where you're likely to have been many times before your current instantiation. Places such as that are where you will inevitably reinstantiate many more times in your future. This is instantiation. This is life. You and I and your pet otter, every insect, every cell, and every organization of cells, all life anywhere in existence instantiates by this mechanism. While each cell entangles at a unique QEF, a few specialized cells in complex organisms, called entanglement cells, or EC, have evolved to heterodyne, or combine their own unique QEFs. This combination of distinct line channels entangle metamatter at yet a different unique QEF, called a composite or emerged QEF, thus instantiating the emerged individual, you. This composite degree of freedom, called the QEF, together with the metamatter it entangles, is called the life ID. No memories or behavior of the host body is carried or transferred by the life ID. In nature, such properties are electromagnetic manifestations of the host species or vessel only. The closest cultural meme to the life ID come via religions throughout human history, having referred to this, using one word or another, as the soul. Once any QE connection is terminated by sufficiently disrupting the cellular component or inducing death of the host vessel, the previously entangled metamatter becomes available for entanglement by other cells. However, this particular metamatter has been imprinted to some extent by its previous entanglement. Each generation of entanglement, each instantiation, each life imprints information from both the host and QEF to its entangled metamatter. The degree of this imprinting is yet to be determined. This time-dependent, perishable imprinting of cellular state in metamatter becomes available to future cells that entangle this metamatter while simultaneously limiting its entanglement opportunities to cells of matching state. The passage of time decays the imprint left on metamatter, causing a return to a state best described as stem metamatter, to be discussed later in this volume. This transfer of cellular state information may impact cellular behavior and development, and to the extent that this imprinted information manifests an advantage for the cell, may provide a survival benefit. This is the evolutionary mechanism used by early life that predated the development of the DNA and RNA molecules. With QE communication, ergo life, the protocell became the laboratory of evolutionary innovation we see today, from which emerged a great many useful cellular structures and processes, but most pivotally, a clear benefit to augment the cloud storage mechanism of metamatter with a more local, more expandable, and flexible information storage mechanism, which became RNA and eventually DNA. This was the birth of the modern living cell. Much is yet to be learned, but the implications of this process are vast and pervasive. The degree to which metamatter is imprinted by its entangled host and unique QEF will determine, after deinstantiation, or death, the likelihood that your imprinted metamatter will, for a time, reject entanglement opportunities from dissimilar host cells, of even your same or similar species, in favor of entanglement with cells that contain your familial DNA. These are cells that are more compatible with its imprinting, thereby increasing the probability of reinstantiating you into your former family line, or, if less finely imprinted, to any random line in your previous species, or, if less finely tuned still, to another species entirely. Longevity may be a factor in this regard. Also, when we discover the entanglement molecule in nature or within the cell, just as we eventually discovered the DNA molecule in the cell decades after Darwin presented his theory of evolution by natural selection, likewise, this may allow us to develop technologies capable of detecting and tracking each individual's unique QEF in this life or across multiple instantiations. This will change the world. At the very least, it will change the way we write our wills. As for practical implementations, discovering and using metamatter could change everything. Metamatter satellites would be very different yet similar to regular orbital satellites. Even though they will reside outside of this space-time, they'll permit instantaneous communication with any point in the cosmos. This will forever alter the human relationship not just to each other, but to all living creatures, biological or otherwise. Also, for the first time in human history, we could begin to take practical actions in life that would affect the individual's reinstantiation prospect into one's next life, thereby tailoring your next instantiation ahead of time, minus the mysticism and ideology.